physics. He received the PhD in electrical and computer engineering from McGill University in 2011, working with Professor Jenemy Cooperstock on haptics and virtual reality technologies. From 2011 to 2012, he was a postdoctoral researcher working on haptics with one of our co-organizers, Professor Vincent Hayward, at Sorbonne University in Paris, France. Prior to joining UCSB, he was assistant professor of EC at Drexel from 2013 to 2015 in Philadelphia, and he received his MA and BA degrees in physics from UT Austin and Wesleyan University, respectively. Jan, it's my immense pleasure to have you speak in our workshop. The floor is yours.
Well, thank you so much for organizing such a stimulating workshop that I know many of us here near to me appreciate very much. I really like the intersection of topics presented here, and I'm going to go ahead and share my screen as soon as Microsoft permits. Wants my fingerprint, so I think we're okay. Let's see if this works. Screen. And is that working?
Yep.
Great. Okay.
Great. So
just holler at me if you can't see what I'm talking about for some reason. But yeah, because of the really interesting intersections between the topics of the workshop and the research we do, it was actually a little bit difficult for me to figure out what to talk about today. In our lab at UC Santa Barbara, we work broadly on haptics, aspects of both science and engineering of devices, addressing the sense of touch, on new methods for soft electronic design and fabrication, including stretchable circuits I'd love to tell you about another time, and soft robotic devices based on textiles, elastomer devices, and other things like that. But today, I'm kind of the core of our work and of what I'm going to talk about today concerns human haptics. And what I really love about haptics is how it is at the center of such a remarkable range of human abilities, of interacting with the world, perceiving, manipulating, and we can even learn to read with our skin, which is pretty remarkable. These kinds of tasks, the amazing diversity of things that we can accomplish using our manual abilities, are facilitated by a wealth of resources in our body that start with the skin, our largest sensory organ, and its rich mechanics. Thousands of cutaneous tactile receptors that transmit information to the brain as neural spikes along thousands of tactile neurons, proceeding to specialized areas of the brain where this information is processed, coordinated with other senses, but also with motor activity in order to enable all the remarkable activities we can perform. I kind of wanted to start by connecting some of what I'll talk about to robotics, and it was a nice opportunity here to think about some of these ideas. So, grossly generalizing, in many robotic hands, one has within an order of magnitude, similar number of sensors and, say, kinematic degrees of freedom. Right, and there are many examples that one can point to there that are probably familiar to the audience here, where the number of, say, tactile sensors or haptic sensors, position sensors, what have you, are within an order of magnitude similar in your numerosity to the number of kinematic degrees of freedom. What really struck me in thinking about this workshop topic was how for most animals, and maybe most easiest to think in terms of vertebrates, the number of sensors in this respect is actually far larger than the number of what we would think of as, say, analogs of rigid body degrees of freedom, so skeletal degrees of freedom or something like that, articulated degrees of freedom. So, taking the human hand as a case in point, we have, on the order of 10 to the 1 kinematic degrees of freedom, something like 25, and there are lots of caveats there. Whereas in a single hand, one has on the order of perhaps 10 to the 4th tactile receptors and a few other sensor types there as well that are relevant to the sense of touch, and here implicitly omitting some other sorts of sensing that go on as well that are a little bit less relevant to things like manipulation. But if you look at this picture in greater detail, these what one might think of as the actuation portion of this system, comprising skeletal muscles and vertebrates, really are not unitary. They're comprised of numerous motor units, which themselves are each arrays of muscle fibers innervated by motor neurons. And groups of these motor units cooperate as a heterogeneous motor pool within a single muscle, and then spanning these units at different layers, as well as multiple muscles. One can really profitably regard these capacities, these specializations, as collective degrees of freedom in a synergistic hierarchy. And it's kind of a rich array of research over the last couple of decades that really views these collective degrees of freedom as being instrumental to control and also reflected in kinematics. And so, this perspective on the motor system has been really fruitful in both human sciences and in robotics. And so what occurred to me as an interesting question is, whether analogous organizational schemes underlie human haptic sensing that might help us to understand why humans have so many tactile sensors in the hand, for example, and whether these might provide useful guidance for robot engineering. And I'll return to a couple of these ideas during the talk. Being engineers and not neuroscientists, we started with a relatively simple model. This is an array of we've designed a few devices like this. These are wide bandwidth accelerometers that can be worn on the hand and emit hand function. They allow the hand to perform articulated movements while the hand is contacting objects in the world, interacting with the world, and things like that. We capture a wealth of information reflecting how different locations on the skin vibrate. The mechanical signals, actually typically transient, like you can see here during natural hand interactions. And one of the things we've done with these instruments is to collect data from the skin, reflecting skin oscillations produced during natural hand contact interactions. So contacting with individual digits or grasping objects or touching objects of different materials. So this yields data at each of these sensors. And we combine this data to get a more integrated picture using some local physics, coupling the tissues at different locations. The data looks like what you see on the right, which are reconstructions from these pointwise measurements with a little bit of physics. And on the left, you can see a reenactment of the gesture that's performed. The thumbnail videos on the right are slowed down, but what you can already perceive from these is that the simple act of tapping 1 or 2 digits elicits mechanical signals that readily propagate throughout the soft tissues of the hand. If we analyze this as we've done according to the action that was performed or the object that was touched or how an object was grasped, what emerges is a really rich palette of sensory information available to the hand, to the soft tissues of the hand. This wealth of sensors we have in the hand that reflect where we're touching an object, how we're touching an object, to some extent the materials involved and things like this. So for example, while the display range here is sort of limited by our visual modality. What we can see from the data is that the simple act of, say, tapping a surface with a single digit elicits prominent oscillations throughout the hand, including adjacent digits. We've used this data to ask some questions about, from a mechanical standpoint, about what sorts of information might exist in these what constitute propagating waves in the skin, as will become clear in subsequent parts of my talk. But we've taken this tactile data set and applied an optimal encoding method to infer a compact lexicon of primitive wave patterns that can more efficiently represent these, nominally continuous degrees of freedom that we've captured through these data. I won't go into all the details here. They're described in our paper, but we take these skin oscillations captured throughout the hand and apply a minimal encoding model in which we take unlabeled data and estimate both compact lexicon of representations and time-dependent activations or weights for each, and do so in a way that minimizes residual error. And these primitive wave patterns that emerge from this capture most of the information in this data set. There's a couple of different ways that statement is made rigorous through our work. But the structures that emerged are quite interesting. They vary in both space and time, but have a localizing quality in many cases, also fade rapidly. And this is linked to the physics of biomechanical coupling in the skin that causes higher frequency oscillations to decay especially rapidly, and well, generally oscillations at all frequencies. They exhibit organization from greater to less individuation. So they tend to be localized in individual digits, initially at the ends of the digits, and spread throughout the remainder of the hand in a way that would be predicted from wave mechanics for contact felt at the fingertips, areas that we often contact objects. Parenthetically, this sort of organization from greater to lesser individuation here is sort of opposite to what one sees in motor studies, where in, say, kinematic or postural synergies, one typically sees organizations that proceed from less to greater individuation as we proceed from kind of less important basis elements to more important ones. They emphasize digits that we use more often, and it turns out we get similar results from various dimensionality reduction methods that we've applied, anything from kind of deep learning methods to matching pursuit methods, and the qualitative findings are all strikingly similar. Go back. Well, I guess what I was going to say here is only that one way to see that these capture most of the information in the original data set is that we can achieve accurate classification in this lower dimensional space, actually on par with what we obtain using the entire data set. When the actions that elicited these vibrations are expressed, the vibrations are elicited by those are expressed through these basis sets. We can often associate them with relatively sparse patterns of activation. I'm sort of presenting a simplified picture of these attributes in a time averaged manner here, just for efficiency, but these conclusions hold true in a time dependent analysis as well. There we go. Okay. And another thing, here, referring back to our kind of raw data and the vibrations in the hand that are produced through different kinds of actions. One aspect of these data that really stood out emerged in the frequency domain, where we could see that there was structure not only in time and space, but also in frequency in a sense. And one characteristic of these data that we found kind of interesting was how the largest extents of skin vibrations really occurs at low frequencies. And this would be expected from mechanical considerations that imply that specifically frequency-dependent damping implies that higher frequencies will become attenuated as these signals propagate away from their source. And so we elected to take a closer look at this in some subsequent work using another measurement method based on scanning laser Doppler vibrometry. So we captured oscillations in the skin throughout the hand here, shown, I think, on the volar surface of the hand or the palm-facing surface, but we've done this on both sides of the hand, and the findings are pretty similar, and consistent with the description I gave a moment ago, namely that low-frequency oscillations readily reach very large distances throughout and beyond the hand itself, whereas higher frequency oscillations become more confined to the location at which they're applied, here at the distal end of the index finger. And there's a kind of an attractive analogy here. If one's interested, for example, in what sorts of information these signals might convey, or what role they might play in touch perception, the ear is a really well-studied model. And within the ear, we have the cochlea, which contains basilar membrane that is coupled to auditory neurons. And the basilar membrane, if you unravel the cochlea into a linear path, has an interesting attribute of segregating frequency according to position or place along the basilar membrane in a way that facilitates early auditory processing, enables different hair cells and auditory neurons at different locations innervating this basilar membrane to respond to signals of different frequencies. So higher frequencies become confined closer to the base of this membrane, whereas lower frequencies readily travel toward the end of the membrane. Here we're talking not about the basilar membrane, but the skin, and a few lines of evidence indicate to us that the signals of interest, these vibrations in the hand, travel primarily through soft tissues of the hand. And an appropriate physical model for understanding this process really is that of elastic waves. So elastic waves are governed by an elastic wave equation, here describing free evolution of elastic waves. And in the presence of damping, which is pretty prominent in the skin because of the large aqueous component in biological tissues, damping plays a significant role as well. So if we look at, say, a plain harmonic wave component of a solution, we have both this oscillation component, but then a damping component, which typically depends on frequency in a manner that causes the amplitude of these waves to become damped with increasing position from where they're produced, but also to do so in a way that's more pronounced for higher frequencies. So reflecting the qualitative findings I pointed to earlier. Here on the right, you can actually see, as a function of distance and time, what this looks like. So in these data captured using the laser vibrometry system I showed a moment ago, you can see that there's a prominent spatial waveform that evolves through time, and that at high frequencies, the amplitude of this waveform becomes rapidly diminished with increasing distance, whereas low frequencies remain relatively prominent in amplitude to large distances. And assembling data from many subjects, we find indeed that the distance to which sort of nominal distance, say median extent here, to which these waves travel decreases sort of consistently with frequency. With some variations here in the data that may reflect aspects of the hand morphology and composition as it varies along the length of the skin. An interesting question for us was also whether these effects are actually relevant to perception, because relatively little on the side of haptic perception has accounted for these processes until relatively recently. And so in kind of one fruitful demonstration of how indeed these are relevant to perception is sort of illustrated here. As engineers, we after some experimentation, we developed a set of stimuli for which the frequency extent varies systematically. It either decreases monotonically or increases monotonically. And in behavioral experiments with humans, what we found is indeed presented this way, naive subjects very consistently report these what they feel in a manner that's consistent with our what we would expect from mechanics, namely that low frequencies have a large spatial extent, higher frequencies a lower spatial extent. So here the sequence produces a pretty consistent perceptive expansion or contraction in the hand. It's kind of a new haptic perceptual effect, and kind of captures one thing I like about working on haptics quite a bit, which is that often if we discover something new, we can come up with a relatively robust demonstration of the effect that anyone can feel. Kind of the last topic I wanted to talk about briefly, just to wrap back to the question I posed toward the beginning of the talk, but also some of the content earlier in this workshop, is one we've been thinking about recently, which is roughly why are there so many tactile receptors in the human hand? So again, we're talking about on the order of 10 to the 4th tactile receptors, which is quite numerous viewed from a robotics perspective. We've been looking in particular at Pacinian corpuscles, which are tactile receptors that are numerous in the hand. It's actually not known exactly how many there are, but the number is certainly within an order of magnitude in the range from 10 to 2 to 10 to the 3. So you can see kind of a map here reflecting more or less the distribution of these Pacinian corpuscles across the hand. Individual Pacinian corpuscles are exquisitely sensitive. So these receptors respond to really minute transient or vibratory inputs that are felt anywhere in the hand. So at their frequencies to which they're most sensitive, we're talking about amplitudes of skin oscillation on the order of tens of nanometers. And they'll, as a consequence of their sensitivity in the biomechanical coupling in the hand that's reflected in the data I showed before, they'll essentially respond to minute tactile signals applied anywhere in the hand. There's another element to this story, which is that essentially all PCs exhibit grossly similar response characteristics. So they don't encode amplitude via rate code. Rather, they phase lock to stimulus frequency over quite a wide range of frequencies. And the variations in their spiking response characteristics are very weakly with amplitude, in fact. So put together, it really calls into question why we would have so many of these, because since they tend to respond to inputs across the great majority of the hand surface and have similar characteristics, naively the information they collect would seem to be quite redundant. And I don't know that we have an answer to this yet, but we've been working on trying to understand what's going on using methods drawn from those that I described a moment ago in tandem with some recently developed models of the spiking response of PC receptors themselves. So we capture with pretty good spatial resolution and temporal and very high temporal resolution vibratory responses on the skin in response to stimuli supplied at different locations here at the index fingertip, and capture the veridical skin displacement or velocity at each of these locations. And after some work, we've managed to couple that to a population model of Pacinian corpuscle receptors throughout the hand. And the results look sort of like what you see here at the bottom. So for the subpopulation of receptors along this axis shaded in red, we get for each receptor oscillation waveform in response to a supplied fingertip vibration. And this neuron model then allows us to predict spiking responses from each of these neurons in response to the stimulus here that was applied at the fingertip. While these results are preliminary, our analyses to date suggest strongly that this biomechanical coupling that facilitates these transmission of vibrations as waves throughout the hand has the function of diversifying information captured from these populations of nominally rather similar in response characteristics receptors. Our results show that the frequency selectivity of receptors across the hand varies markedly. Patterns of variation are somewhat complex and interesting, but one thing one can see from this is that after biomechanical transmission, the frequency selectivity of these PC receptors varies quite a lot, and a further finding from our analyses is that if we look at the correlations between these spiking signals from different receptors, then in taking into account as we have the mechanical signals transmitted, the mechanical processes transmitting these signals throughout the hand from where they're applied, we see much less correlation between individual receptors than would be predicted from a simplified model, accounting only for, say, distance-dependent damping, which is, I think, integrated in the touch sim model of Visell and all. So we're still working on this, but intriguing evidence for the idea that the soft mechanics of the hand really plays an important role in tactile sensing by diversifying information from these populations of similar receptors. And plausibly, this helps us to understand why we might have so many of these receptors. Well, they seem to be capturing far different information than would be expected in conventional descriptions. I want to thank you again for giving me the opportunity to talk and really acknowledge the work of all the outstanding researchers that we have in the lab at the moment, and those who have been through our lab in the past years. I want to acknowledge our funders, especially the National Science Foundation, and thank you again for the opportunity to talk with you here today.
Thank you, Jan. In the interest of time, maybe we'll just take one question. Is there any question from the audience? Yeah, I see a raised hand. Oh, Lynette, yes. Please feel free to unmute yourself.
So, Jan, I just have a question about your last comments to do with the number of mechanoreceptors in the skin and so on. The estimates that people come up with tend to be based on Johansson's data from the 70s and 80s, in which he looked at tactile afferents and peripheral nerves. And there's an assumption that there's sort of some one-to-one mapping between those afferent fibers and mechanoreceptors. And